Hello class. As you can see on day 4, for me, which is June 7th, because I planted these on the 3rd, there's a lot of plant growth. These are genetically modified, so it's not typical that in 4 days you'd have this kind of plant growth. Also notice that there's not the same amount of plants growing in each hole within the cell. I want to make sure I'm defining those terms again. Each of these is one hole, and the styrofoam cube is the cell. Now, if you followed the instructions that I gave you, you planted more than one seed in each hole. And if you didn't, it's really not a big deal. I want to impress upon you at this point that this is much more about the process than getting a good data set. Some of you will not get good data sets. We do something similar in our face-to-face classes, and every semester there are some students that, even though they've followed all the instructions, they don't get a good data set. And there'll be a lot of us who are getting data and we can share. So please don't be too concerned if this isn't working out for you the way you were hoping. In any event, there are more than I planted more than one seed per hole in each of these treatment cells. Some of them have 2 growing, some have 1, some even have 3. At this point, what I want to do is make sure each of the holes have only 1 plant growing. And today I'm going to take my first data point, doing something similar to what you see here in this image. You may also, before you start thinning the plants, you may also want to make some anecdotal observations today about what you think. Like, for example, in my control, I have the most plants growing, even though I put the same amount of seeds in each of the holes in each of the cells. I planted 3, I believe. No, I did. I planted 3 seeds per hole in each cell. My recommended amount of fertilizer doesn't actually look like they're quite as tall. And the tallest plants I have are the double dose of fertilizer. Going back to my hypothesis, I thought if fertilizer were added in the proper dose, then the plant growth rate would be the fastest over the time that we're doing our study. Anecdotally, it doesn't really look like that, but we have no data. We just have an observation looking at them. So now it's time for me to thin these out so I can start calculating data. This is a little bit tricky. This was direction number 14 in the paper instructions that I gave you. So today, I'm going to show you how to do number 14, thinning the plants, day 4 or 5. I'm going to be doing a method like this, very carefully plucking out all of the plants in each of the wells except for a singular plant in each hole. How am I going to do that without biasing my research? This is a first really important question. A bias can take place if I think about what I expect and then I pluck the plants to reflect what my bias is. I hope that makes sense. We call that cherry picking data, and it's not sound science. So to have a more random approach, I'm going to eyeball the plants in each hole and take out whichever one seems to be intermediate. And if there's only 2, I'm going to try to choose at random the best ones to remove. This is never going to be perfect. but we're just going to do the best we can to avoid bias. I also would suggest that as you carefully pull some of these plants out by the roots, you may want to set up a secondary little pot like this one with some soil and transplant the ones that you pull out into this, just in case you have a lot of mortality, meaning just in case a lot of them die, you may have some additional plants to try to gather some data from. I'm going to go ahead and stop this video and get set up to show you how to actually remove the plants.